So the last thing that we were looking at was the Hardy-Weinberg Equation and how it relates to population genetics. And so this equation essentially says that the frequency of one genotype here plus the other genotype, so this is the homozygous for one allele, the heterozygous, and then the homozygous for the other allele, that the frequency of those genotypes in the population would equal 1, or 100% of individuals in the population would have one of those possible outcomes. They'd either be, say, homozygous dominant, homozygous recessive, or heterozygous. And that's kind of the idea. And then we said that if we were to then calculate the actual frequency of these alleles over time, that the Hardy-Weinberg principle states that in equilibrium, the frequencies will not change. Okay, so here, in this little example, we have generations. Generation 1, generation 2, generation 3, and then we have the frequencies of the individual alleles, right? In this case, P or Q. And so one of the examples we were using was for flower color. So we had C for color, R for red, and that was P. And then we had W for white, and that's Q. So we're seeing that generation to generation to generation, it's not changing. And so this population is in equilibrium, which means it's not evolving. So what's required for that circumstance to occur, for things to not evolve? Well, these are our conditions here. All right, so these are the conditions that must be met for a population to be in equilibrium or not evolve. And I'm going to go through each of these. Some of them I'm going to talk about more than others right now. Some of them, this will be all that they're covered. They're just going to be kind of mentioned and defined, and that's pretty much it. Others we're going to spend more time on and go over in other aspects of the course as well and in other lectures. So the first one is no mutation. Okay, so mutation can't be occurring within the population. If it is, then obviously that would change the alleles or allele frequencies. So how? And again, without going into all the chemistry and molecular biology of it, I'll give you an example. So let's say we have a segment of a gene. Obviously not going to be a whole gene here. Here's the sequence: A, T, C, C, A, C, call that a G, A, T, T, A, C, A, like this. Now when DNA replication occurs, two strands of DNA are pulled apart, one strand acts as a template, and then a new strand is built, and there's all sorts of rules about the directionality and the enzymes that do it and the energy required and all these sorts of things. But a new strand will be built. We're using this one as a template. T's pair with A's, G's pair with C's. So there's certain rules. So A and T must pair, G and C must pair with one another. And so as we go through like this, let's say we put a T here. And we continue this, T, G, T, like that. And we have this new piece of DNA, the blue one that I put here. We look at it and try to follow up. Do the rules match? A's with T's and G's with C's? What we'll find is that there's a place here where there's been an error. Okay, so this is the new strand of DNA. There's a T there. T's are supposed to go with A's, but this is a G. So technically that should be a C. If that occurs, and that sort of thing does occur all the time, there are random mutations like this that are always occurring. Cells have machinery to proofread, just like spell check, and check after the fact and say, oh, look, there's a mistake here, and let's fix it. And often it is repaired. Sometimes it's not repaired, and sometimes it stays. And so if it stays, then what happens? Well, that alone then is a mutation, okay? And it's called, get rid of this wicked right there. It would be called a point mutation. It's happening at a single point. And what that's going to do is change this information here that's been copied. So now these three nucleotides generally are going to code for, well, they may not code for amino acids because it might not be messenger RNA, but that's kind of the idea. Is they code for something, and if the information is wrong, the information that's coded here isn't going to do what it's supposed to. So the piece of RNA isn't going to function, or the RNA is not going to give us the information for the correct protein, or an amino acid might be substituted, or so on. These point mutations can be what are called silent, meaning the switch here doesn't really do anything. The result, maybe this is an area that doesn't code for anything, so nothing really happens. If it's an area that codes for something important, maybe it codes for the same thing, because if we study the codons that code for amino acids, what you'll eventually find is that there are 20 amino acids but 64 codes, which means that many of the amino acids have more than one code. So you could switch one of the codes to one that codes for the same thing, and it doesn't really change anything, so nothing really happens, even though technically a mutation occurred. Or you can have a substitute, so you substitute something there, and then you get something called a missense mutation. And that's where we get a new, maybe, amino acid. A different amino acid is coded for, and that could be maybe not change much because they're both polar amino acids, or changes a lot. The nonpolar amino acid behaves very different than the other, and the protein doesn't function. So one single amino acid change could have a drastic effect, or it may have very little effect. In addition, there's even more drastic things that can happen. So some of the codons that are coded here code for a stop. So essentially for the code to say stop reading here, like don't read anymore past this point, and the mutation could change this one into one of those stops. Right? So that becomes a nonsense mutation. Tells it to stop reading, and so now that's really going to be functionless. So these are types of mutations. And then there's even more dramatic ones, which instead of a point mutation here where we put just a wrong thing in, nothing is put there. It's actually a deletion. So at that point, all this information is going to have to then shift over, which means everything that was supposed to be coded for in these groups of 3 is now going to be altered by 1. Kind of get this idea. And that means this one will be wrong, this one will be wrong, the next one will be wrong, the next one, everything from that point forward is going to be wrong. Okay, that's called a frame shift. Right? And again, this is something you don't actually have to know that right now. I'm just giving it to you as an example. And that frame shift then causes everything to go wrong. So these are normal things that happen, and what we're saying is that can't happen. If that happens, then we're going to change things like some of the alleles and the allele frequencies in the population. We could duplicate things, and all sorts of other things can happen. But for a population to be in equilibrium, none of that is going to occur. Which then means, obviously, for evolution to occur, some of this doesn't have to happen, but it could be happening. That's kind of what we're going to get to next, just sort of what happens to change the frequency. So random mating. So that essentially means there's no selection. And this is selection of mates, referred to as sexual selection. So you see in birds, right? Male birds often are very colorful, and the females are more drab. So a cardinal, red cardinals that you may be familiar with, are the males. The female cardinals are more brown, orangish color, right? So that coloration is a signal to the females, maybe that they're healthy, and that they might be more attractive to the females. And so that would be related to sexual selection, and different colors or brighter colors or changes in feathers. Birds display a lot of this type of thing, is involved in the selection of mates. But with random mating, there can be no selection. Right? It has to be completely random. So the example of this would be spawning in marine invertebrates. So a lot of marine invertebrates, say like a sea star, is moving along the bottom, and that's a bad arm. A signal is often sent out from some sea stars, and they often cause an aggregation. So a whole group of them may come together, and this signal then triggers a release of their gametes. So they'll start to release just gametes into the water, and so sperm go into the water, eggs go into the water. And fertilization occurs. No individuals are mating with one another. It's called a broadcast spawning event. And so there's no selection going on. There's no, in terms of mates, all of them are just putting out their gametes into the environment, into the water column, and fertilization is completely random in what's occurring. And so that's a type of random mating. And there's other examples of that as well, but that would be one that is very common for some organisms that actually have that particular style of reproduction. So that has to happen for it to be an equilibrium. If there's selection, if one sea star chooses one partner over another, then this wouldn't be true. And then we might change the frequencies, but in this particular case, we're saying, nope, there's no choice in the matter. They just all kind of randomly go together. Natural selection. This is one we'll talk about as a much larger topic as well with Charles Darwin. Before this as well, I'm talking about Darwin and how he came up with his natural selection as a mechanism of descent with modification or evolution. Essentially saying that there are no environmental factors causing any kind of stress. There's no nutrients that are being competed over. Essentially, there's no competition. There's not really the predation going on. There's no real selection. Or if there is, it's happening equally to all individuals of the population. So whether it's plants or fungi or animals or single-celled organisms, that if some are killed or eaten, any individual is equally as likely. Larger ones, smaller ones, ones of this color or that color, it doesn't matter. Completely equal in terms of success or failure, essentially, in dying. Now, natural selection, what it's also about is fitness, which is kind of related to the sexual selection. Now, people think about this, and the explanation is often off a little bit. Maybe not necessarily wrong, but it's sort of often very incomplete. So people talk about survival of the fittest. So talk about survival. And when we think about survival, think about individuals being stronger maybe than others. That's not really what it's about. What biological fitness is about, it's not about pull-ups or anything, running fast or anything like that. It could be related to it because it could lead to survival in a certain situation, but an individual surviving has nothing to do with population genetics or the frequency of alleles in the offspring. See, fitness has to do with offspring. It has to do with putting genes from one generation into the next. So obviously, if you die or are eaten before you reproduce, your fitness level is 0 because you have put no offspring into the next generation. Individuals who put a lot of offspring into the next generation are considered very fit biologically. Right? So in terms of natural selection, what we're saying is that no individuals also have a better chance or worse chance of putting individuals into the next generation, right? Because they're not being selected against by the environment, and they're not being selected for preferentially than any others by mates as well. So there's no sexual selection, there's no natural selection of the environment selecting them. And the population is very, very large. So the next characteristic, we have to have an extremely large population. And I'm going to give you an example in a bit here of what would happen in a small population where things can very randomly change. Okay. And I'm going to actually, that's a first big example I'm going to give you. And so I'm going to just hold off on it right now and just talk about the last one. So we're going to talk about this in just a second, just a little bit more in more detail. Obviously, a large population going to be much more random mating and that sort of thing. Whereas a very small population, it's a lot of things could happen, and only certain genes might survive to the next group, or the types of mating that occur might not be as random because you don't have that many individuals. So we'll go over some very specific examples of this in a second. And then the final one here is no gene flow. So for a condition for Hardy-Weinberg equilibrium to be in place is that no new individuals can come from other populations. The population is just set. And we can't lose individuals because we can't have individuals leaving the population to go someplace else, because then we lose their alleles, and then potentially the frequency of alleles may change over time. So what happens then, if, we get something like this that happens. So all of a sudden now we look at the next generation and there's a change. So here we have equilibrium. And now in this generation, we see, whoa, the allele frequencies, they're not the same. There's some change. And this is what we refer to as microevolution, a change in allele frequencies over generations. And then we could keep following this more generations to see, does it stabilize here at 7.3? Does it go back to 8.2? Or does it continue to change? Do we keep getting these sorts of changes? And so to give you an example, of kind of how that would work, let's just say we have a very, and this is also in your book as an example. So let's just say we have a population of the flowers, like we were talking about before. And let's say we have, I'm just going to use some letters here and do sort of the color. So let's say we have red flowers, the pink flowers, and the white flowers. And let's say we have a very, very small population of just 10. So in our total sample here, our population is 10 individuals. Okay, so we're going to have a red, a pink, a red, a white, a pink, I think this is, red, pink, another pink. So we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so 10 individuals in the population, and that's all that we have. And let's say that in this population, we have a red, a white, and a pink that reproduce to go into the next generation, and then 2 of these pinks go into the next generation. But these others, for some reason, whoops, this pink does, they don't mate. The idea here, you don't have to keep track of which one is which, just the idea is this. In a very small population, what could happen is an animal can step on some of these flowers and kill them, and then that's it, and then they're gone, and they're not going to reproduce and pass on any genes to the next population. And very randomly, they could step on all of one color, just not on purpose, they just happen to do it, and then they're gone, and then those alleles are just gone from the population, and they can't go on to the next generation. So it's a small population to begin with, so we take away some individuals, we lose some, and now it's going to be changed. So let's say this one had a p-value of 0.7, and the q obviously is going to be 0.3. Now in our next generation, because we had more pinks here, and then the red and the white, we have a change in the population. And so we're going to get the P being 0.5 and the Q being 0.5. And so now on this one, we have white, red, pink, white, red, pink, white, pink, pink, red. 1, 2, 3, 4, 5, 7, 8, 9, 10. So we have 10 more individuals in this generation. So again, we're keeping it small like this. And let's say the same sort of thing happens where, certain individuals die and other individuals survive. And maybe in this generation, only these red ones actually have offspring. That's it. Now remember, the reds, there's no hidden other allele. If it's red, they are homozygous for red. That means all 100% are going to be BRCR. So P is going to equal 1.0, Q would equal 0. That allele's gone. It's like lost in the population. And over time, it's not going to come back. It's just gone, right? And so you'll never have pink or white individuals again in that population. That's an extreme example of a very tiny little population, but that's the idea here. Why, we need extremely large populations to maintain this, but it's also an idea of how, as populations, for some other reasons, which I'll give you like actual reasons. So this is like somewhat of a silly example, and that you're not going to just have a whole population probably of 10 flowers of a species in the wild. Although some organisms, as they become endangered, do have very low numbers, in the wild. But some of the things that happen related to this would be a founder effect. So let's say this is an island, and this is, you know, a bigger island or a continent or something like that. And we have organisms that live, you know, here. And there are none on this island. And a storm or something now transports a few individuals over here. So very few individuals. They are the founders of this new population. It's unlikely that their genotypes are going to be a complete representation of the original population, right? They're not necessarily going to be transported in the same numbers. So whoever happens to be the founder, if they were, say, in the flower example, more white flowers or pink flowers or the red flowers, especially if they were the red or the pink more so, that's going to dramatically shift the allele frequencies within the population. So they might just have only white flowers, say, on here, because the founders were only white flowers that got transported over here, the seeds from the white flowers, that sort of thing. And so the founder effect is an actual thing that happens where islands, you know, say, for example, are colonized by new organisms, and those organisms don't represent fully the original population, and that's one of the things that then drives speciation, which we'll get into, is how the new species form from other species because they are genetically different, and then they become geographically isolated, and then over time they change, and they change dramatically. And then sometimes maybe there's new conditions on the island, so then other things like natural selection actually will occur, and then we're going to actually have selective forces working on them, different forces than that are in the other habitat, and so then we start to see more and more change. There's also a bottleneck effect too, which is the same sort of thing where essentially, you know, a few individuals make it from the one population, the larger population, into some other area, smaller population, or over time, some individuals go through, and then it takes a lot of time for other individuals to go into this new habitat or environment, and the first individuals who go there, again, don't represent the original population, so that population starts to become very different than the original, and you end up with two different populations of the same organism that they may not look anything alike, or they might start to behave very different, and they may over time even become isolated from one another reproductively, where they can't even reproduce with each other. And so those are all things that will lead to speciation, which is what we're getting into next, kind of not directly, but in a few lectures past this. All right, so this is essentially just going over. So the last part of, well, not the final last, we're going to do one more thing on working out a couple problems, right? But we introduce the terms of Hardy-Weinberg Equation about alleles and allele frequencies and how some of the numbers are actually calculated and how we look at that. How that if we have those numbers and monitor them in the population over time and they do not change, the population is in equilibrium, which means it's not evolving. For that to occur, we have to meet certain conditions. There can't be mutation. Mating has to be completely random. There's no selection of mates. There's no natural selection by the environment, the competition, or anything happening between the organisms. They're all equally as likely to give a lot of offspring and the same number of offspring. The population itself is a very large population, which increases how random the mating would be, and so on, and that there's no flow of genes in or out from other populations. So we have to have those occurring. Now, if any of those things do occur, then we start to get change. So if we have mutation, we might start to change the frequencies. If mating is not random, if there's selection, the frequencies might be changed. We might select for one type over another type. If there's selection, maybe a coloration allows the organism to hide better in the environment so it isn't eaten as much by a herbivore, if it's a plant, or a predator, if it's an animal. And so the ones that are of a certain coloration pattern, they survive more than the others because the others are being consumed, you know, or there's all kinds of other adaptation, physiological adaptations and things that could be a result of mutations or different alleles. Tiny populations like this, because of founder effects and bottleneck effects, things that where only a few individuals are selected from a population or transported to a new location, don't represent the original, and then that new population becomes different. And then if there is gene flow, so if we actually get new individuals coming from a different population, maybe they have more of the white, in this particular example, allele, then that might boost up the number of white alleles in the total population, right? And so that's the thing that could then drive a whole change in that population over time, which we are defining as microevolution. So that's a change in allele frequency over time, is what we're looking at. So the last thing we'll do with Hardy-Weinberg is I'm going to do one more short lecture on problem solving, and there'll be types of problems that you would do in worksheets for homework, and I'm just going to kind of work out one or two so you can kind of see the type of thing that you're going to have to solve, all just using the same basic equation, but this way you'll be figuring out, you know, frequencies of one allele or one genotype or even a particular phenotype in a population and going back and forth in different directions. So using that information to find those things out.